So
Okay.
if we are ready, I am just gonna very briefly introduce Akankshi. Akankshi and I have first met 10 years ago at the embryology course where we were both students, and she had just started her postdoc in Sean Megason's lab, so she had transitioned from Drosophila morphogenesis to zebrafish morphogenesis. And she's now an assistant professor at Duke, and she's gonna tell us about principle of tissue morphogenesis today.
Thanks, Vanessa. Yeah, I have very fond memories of the embryology course. So if there are trainees in the audience who are looking for an experience like that, we both endorse it, support it heavily. Okay, so yeah, I'm sorry I couldn't be there in person today. I'll be there from Monday onwards next week, and I'm really looking forward to having in-person discussions. And just a brief introduction. Vanessa already introduced me, but I just want to emphasize I'm trained as a biologist, and my lab studies principles of tissue morphogenesis using zebrafish development. At Duke, I teach graduate students about basic concepts in physics and math, and also state-of-the-art quantitative approaches to understand developmental processes. But of course, you all are at the Center for Mathematics, so I'm not going to try to teach you physics or math. Maha suggested this to be a developmental biology tutorial, and I'm sticking to that. So I'm going to share how developmental biologists like me think about shape, about building a common language that can link genes, cell behaviors, tissue mechanics with ideas of geometry and topology, which is the main theme in the workshop here. So many of the ideas that I'll mention have somewhat been already raised in the previous tutorials and research seminars. So I might be repeating some of those things to make those points. So just bear with me. But think of this tutorial as mostly if you're an incoming graduate student or postdoc in the field and want to get an overview of how we think about questions from within developmental biology. So I'm gonna have a couple of cliche slides, but they really make the point of um, why shape is so fascinating. So here you have a vertebrate eye, a whole organism, um, and you see some branches of a bonsai tree and a picture depicting roots of Arabidopsis. They're all, of course, evolutionary distant and mechanically different, yet they all solve this problem on how to transform genetic information or information in general that they pack as a single cell into a three-dimensional form. So this is my second cliche slide to make the point. We'd like to start thinking about shape from the beginning, which we attributed to fertilization. One can argue that it's even before when the egg forms, but let's consider this as the starting point. So this single cell here, of course, it's packing information in the form of its DNA, in the form of the genome. But it's also packing other information in the form of where the site of the sperm entry is, the cytoplasmic flows, which you don't really see here, but they've been observed in other species. And you have spatial asymmetries in the form of actomyosin cortex and microtubules when the sperm enters. So it's packing already a lot of asymmetries on what seems to be a symmetric spherical cell. Um, what follows next is the expansion of this single cell to make more such stem cells that have all the information to undergo specialization later on. So in this case, you see a mouse blastocyst where in a few rounds of division, it transitions from the single cell to a multicellular structure with distinct inner and outer inner and outer cell populations that will, and this sort of inner and outer position will determine their fate. You can also see at the end of this movie, there is a lumen-filled cavity that forms that gives more asymmetry to this tissue. So you already see that this starting from this simple single cell, you already form a structure that has some boundaries, a lumen, some polarity. Beyond this point, again, speaking as a developmental biologist, we like to image these processes. It's technically very challenging to image mouse embryos in vivo with most conventional methods. It is possible with light chain and adaptive optics, but on a day-to-day basis, it's extremely hard. And of course, it's hard to visualize this in human embryos with the ethical constraints. So to visualize the next major transition that happens to a blastocyst, we turn to other model organisms like flies and zebrafish. So here you're looking at a zebrafish embryo. It's from single cell to blastula, transforming into an embryo with a defined body axis, with all its three germ layers that will later on specialize, and a recognizable body plan. So I'll let you sort of appreciate this movie because this is really morphogenesis at full scale. So once these axes are laid down, and Olivier did an excellent job in speaking about the body axis patterning, and it complements everything I'm going to tell now, which is entirely on morphogenesis part, not patterning. Um, these tissues here that are still not fully differentiated into whatever functional structures they're going to form will continue to undergo massive morphogenetic remodeling events. So here, these are corresponding to the four pictures I showed earlier, where you saw the vertebrate eye. Here you see the vertebrate eye in the making. This is the optic vesicle that folds inward to form a double-layered cup. It later differentiates into the neural retina and the retinal pigment epithelium. And this concave geometry is essential for spatially organizing the photoreceptors that are required for this optical function. In the second example here, you see a leg imaginal disc of Drosophila, where you see, this is in culture, so you see this leg proper elongating inside this peripodium layer that tears apart when the elongation is happening. In this example, you see Arabidopsis root tip growing. So these have stem cells at the tip that continuously divide. Their daughter cells elongate, and depending on the plane of division, they can form. They can elongate the tube or form these radial layers of the epidermis, the endodermis, and the vasculature. This is another beautiful example of a mouse kidney epithelium undergoing iterative branching morphogenesis. So it creates this very arborized network to maximize filtration surface. So these, I these are very different. The shapes, the functional shapes that they are giving rise to are very different, but they're relying on a surprisingly small set of physical mechanisms to transform these simple tissues into these complex functional structures. And here's a list. It's not an exhaustive list, so if I missed your favorite molecular effector, I apologize. But I think these are one of the key drivers of tissue remodeling. Cell contractility generated by filamentous actin bound with motor proteins like non-muscle myosin or muscle myosin. You can have adhesion either between cells or adhesion between cells and the underlying substrate, the extracellular matrix. The composition of the matrix itself can sometimes drive morphogenesis. A lot of tissues pump ions into their surrounding spaces. And these create osmotic gradients that can lead to influx of water that can build hydrostatic pressure if this is confined within an enclosed tissue. And just the overall growth of the tissue can also drive morphogenesis through just cells dividing or cells growing in size. So in the next few slides, I'm going to try to summarize how these molecular drivers are deployed to generate all those shape changes that we observed. So if the goal is to bend or invaginate, um, so just picking up where I got disconnected. So if the goal of the tissue is to bend or invaginate, we know. I mean, most people in this audience already know this amazing work done by several groups, and especially coming from Adam Martin's group, that show that you can pattern apical constriction. I'm trying to move my slides. So we know this body of work where you can constrict apical surfaces by polarizing actomyosin contractility and sliding junctions on the apical basal surface, and this creates bends in tissue that subsequently invaginate. In another example, it's very different from using contractility. You can deposit a specialized extracellular matrix that's rich in this component, hyaluronic acid, that can osmotically swell. And that can cause the overlying cells to stretch and bend or bud, depending on how many cells are synthesizing this matrix. So next, if the goal is to elongate the tissue, and here again, I'm sure you're familiar with this. Instead of isotropic contraction on the apical surface, you can have planar polarized contractility that leads to these polarized rearrangement of cells or directional rearrangement of cells that drive tissue elongation. And of course, several examples come in mind, but I've highlighted the most famous one of the Drosophila germ band. And in this case, in the second example, you can drive elongation not from patterning intracellular forces, but rather patterning the substrate around the apical matrix around. So if you have a growing tissue wrapped in a stiff ECM, and if this ECM is aligned circumferentially, constraining radial expansion, it forces this growing tissue to grow in the orthogonal direction. And this model has been proposed to explain egg chamber elongation. Again, it's body of work done almost at this point 10 years ago, and it's very well characterized. So now, if the goal is to make a tube, there are multiple physical routes to get there. One mechanism is folding, and this is how a chick or a mouse neural tube would do, where you have apical constriction at these hinges that lead to bending of this sheet of cells, and then you have these ends of this sheet that need to close the neural tube. They make new contacts to close off and seal this into a tube. Another way to make a tube is by cavitation. So here, essentially, you have a mass of cells that reorganize into a tight epithelium, and they start pumping in ions and create a lumen de novo, often through accumulation of fluid because they're pumping ions. And this causes this cavitation and opening up of these cells into a tube. And this is how zebrafish makes its neural tube. Okay. So in another example, if the goal is to expand a tissue without losing its organization, and this is a common feature that happens in adult homeostasis, but it often happens during development. development do. One mechanism of the skin epithelium where if you have the orientation of mitotic spindle in plane of this basal layer, so this is the stem cell layer, it will make more of these stem cells. But if it's perpendicular to this player, then it will really result in stratification of this skin or more differentiated cells. And this way of doing oriented cell division is essentially how plants perform morphogenesis. So plants are, as you know, they're encased in rigid cell walls. They don't really migrate. It's really the plane of division that's largely determining their entire architecture. So here they would follow the cells, because of their cell wall, have turgor pressure, and they follow geometric rules to decide the plane in which they're going to divide. And they can either cause longitudinal extension, the way we saw for the root tips, or they can orient the plane to form those radial layers, which are more specialized cells, either the epidermis or the endodermis. Okay, so another mode that I want to highlight is where the tissue is not bending, dividing, it's just simply being stretched because it's experiencing forces. And this is a clear example comes from a lot of these luminized tissues. And in this case, I've showed how the embryonic inner ear, the aortic vesicle initially expands. It simply pumps ions in over here, where building hydrostatic pressure, and then the surrounding cells, they're not dividing as much. The contribution from cell division is just 10%, but most of this expansion, at least in the beginning stages, is coming from the cells stretching out significantly. And these two examples, again, in CMSA community, I'm sure you've heard from Maha, but I still want to highlight, because a lot of morphogenesis happens this way, where it's not dependent on pattern forces, so to speak. It's arising from pattern material properties. In this case, it's coming from differential stiffness, the differences between the overlying epithelium and the underlying substrate. And the example I've shown here, which happens in many different systems, including the two I've listed here, the gut vilification in mouse and formation of feather buds, is where the underlying layer, which is full of these mesenchymal cells that can migrate, it condenses and contracts through actomyosin contractility and reorganizes the ECM around. And this mesenchymal cell is connected to the overlying epithelial layer, which is relatively stiffer. It's mechanically coupled through the basement membrane. So the overlying stiffer layer resists this shortening of the underlying layer. And this mismatch in mechanical properties generates compressive stresses within the overlying tissue. And once they go beyond a certain threshold, it results in the epithelium relieving those stresses out of plane, or buckling, essentially, into buds or folds, as you see in this context. You can also have differential growth. It is conceptually similar to stiffness, but here, developmentally, instead of patterning the material property of the tissue, you're patterning the growth rate. So if you mismatch the growth rate of two tissues, you can have buckling, again with the same principle, where this tissue experiences compressive stresses because it's growing in this constrained environment. And this is, again, like a lot of our understanding comes from the work done by Maha in collaboration with Cliff Tabin on showing how this pattern expansion under constraint can cause these very reproducible organized folds during gut vilification in chicken. Okay, so I've given a lot of examples of morphogenesis in terms of bending, stretching, buckling, but one mode that I want to highlight, and there are many such other examples, but I'm just highlighting one, is sometimes developmental change is something very fundamental about the cell itself. So here in this case, cells can change their properties entirely from being epithelial and sticking to their neighbors. They can become mesenchymal, where they downregulate adhesion, detach from the sheet they were initially attached to, and migrate to places and contribute to morphogenesis of a lot of different organs. And this is essentially how the neural crest, which is part of a lot of our jaw structures, originates. It's coming from this EMT of initially ectodermal derived tissue. And one thing you can appreciate too, that is that these cells are changing their connectivity too. So they were initially in contact with this sheet, and now they change lose those connections and go on to integrate in other organs as they migrate. So, so I think these examples summarize not all, but significantly a lot of morphogenesis processes that we see. And so is the question here that people proposed when we were thinking about the communities thinking about these processes is that how do you pattern them in space? And of course we know the answer is that these are patterned by developmental factors. And this is a developmental biologist appreciation slide for how we started thinking about these genes. So this is in the 1980s and 1990s. Developmental biologists used large-scale forward genetic screens to identify genes for morphogenesis by starting thinking about phenotypes rather than candidate genes, because at the time we didn't know what to look for. So this was done first in Drosophila and then in zebrafish, um, where the males were randomly mutagenized with strong mutagens like ENU, and they were bred for successive generations to find recessive mutations that can be systematically screened under the microscope for defects. And these defects were very apparent. These are defects during gastrulation, failure to segment. We saw a lot about somites, so there were segmentation failures in zebrafish, gastrulation movement defects in thighs, and somite boundaries, neural tube defects, and so on. And what followed after these screens, after generating these hundreds of mutants, was mapping using painless recombination methods and molecular markers to identify the underlying genes. And this was, of course, concomitant with being able to look at the expression patterns of these genes. And of course, we saw these beautiful patterns that could explain how the molecular effectors are being deployed into space. And and Olivia already did a great job in highlighting some of these, the BHLH, the Hox genes. So I would say these broadly, of course, this is not exhaustive by any way, but broadly, the genes that were identified can be categorized into the category of transcription factors, that are proteins that would bind to specific DNA sequences and upregulate or downregulate the genes that are close to them. And the other class are morphogens, which of course you all know about, where these are signaling molecules that form concentration gradients, both in space and time. We don't talk about the temporal gradients of morphogens enough, I think. So these essentially instruct cells to adopt certain phase depending on the dose or exposure of the morphogen they experience. And of course, through decades of very careful and very technically challenging gain of function, loss of function experiments, the community has revealed in remarkable detail what these transcription factors and morphogens are affecting, the downstream genes, their polarity effectors, the biochemical processes that are ultimately driving cell behavior or cell fate decisions. So this sort of gene by gene analysis brought us really far. And then we reached about 10 years ago with this explosive information that we could get from the transcriptome of developing embryos using single cell transcriptomics analysis. So these single cell atlases, uh, maybe Alon will talk about them. I don't know what Alon is going to talk about, but this was again like pioneering effort from Klein Lab and now several other labs, where we've as a community built these atlases across developmental time, dramatically expanding our view of which genes and cell states, how they're evolving over time, over developmental trajectories. We can infer lineage relationships, map gene expression changes during those trajectories at really a high resolution. And and again, this is maybe a bit more recent, but already a lot of advances have been made. Is thinking about transcriptomics in space, so spatial transcriptomics, where in single cell atlases we lose that positional information, but with spatial transcriptomics we can link this identity or cell states to their position to the overall tissue architecture. So this is, I think, it's really remarkable how far we've come in development and in thinking about which genes are important for morphogenic processes. But while these atlases, like even if you computationally order them in time, they're still snapshots and they do not capture the physical dynamics, the feedbacks, the constraints that the tissue experience. And this is something as a community, especially people who are participating in this workshop, this is what we care about a lot, is all these constraints and feedbacks that the tissues are experiencing. And now I'm going to play a time lapse of zebrafish gastrulation again. So you've already seen it from the perspective of the cell. This time you're seeing it from the perspective of gene expression over time. So here, this particular embryo is marked by genes marking the ectoderm, the mesoderm, and the endoderm. And what you'll notice is that as this embryo is undergoing gastrulation, undergoing this large scale rearrangements, the pattern of the gene expression is obviously evolving too. So it's not a static spatial pattern instructing shape changes. The pattern itself is evolving. The landscape is evolving. So the gene expression is changing over time. The context in which the cells are is constantly changing over time. So how do we appreciate this? In essence, the question that we can ask is that beyond the genome, what additional layers of information are required for the cells to organize into a functional shape? So we can think about the first part that I discussed, that you have these molecular drivers spatially patterned by developmental factors as the first principle of tissue morphogenesis. And the second principle can be that we know as a community now, and it's not going to come as a surprise, that of course there are feedback. So information is flowing from genes to cells to tissue dynamics and back. And really, I'm speaking to the choir here. There's just been tremendous amount of work, including our own work that I'll talk about more next Tuesday, where the mechanics, either at cellular level or tissue level, can directly feed back onto gene expression patterns to drive either positive feedback loops or negative feedback loops that drive development forward. So I'm not going to talk much more about mechanical feedback because I'll speak in more detail next week. And really, there's just, I feel like I won't be able to do justice to just the body of work done in this field. Another form of feedback that I want to highlight, which resonates more maybe for the theme of this workshop, is geometric feedback. And it's not entirely distinct, of course, from mechanical feedback, but the distinction here is just subtle, is that where their mechanical forces can directly influence signaling. In geometric feedback, I'm thinking the feedback arises from the shape itself, the curvature, the confinement, the surface area to volume ratio, and so on. So here I've just abstracted using those same cells that if you have if you have a cell experiencing a morphogen around, it becomes it changes shape because of force generation. The concentration of the morphogen it's gonna experience just changes because of the shape itself. So this is an sort of a thought experiment, in that the change in concentration is going to affect the state of the cell by not actually changing the level of production of this morphogen, by just but just of the virtue of changing this shape, or the confining this morphogen into this space. But we can think about it in more realistic scenarios too. So we already heard about Turing patterns. So you have reaction diffusion systems of a short range activator, a long range inhibitor. They interact to generate these remarkable periodic patterns. But those all these class of models are on flat domain. I alluded to in one of his questions to Mattia yesterday. So now imagine placing the same reaction diffusion system on a curved growing epithelium. And again, it's not something new. There are several studies now at this point that go over 10 years old, which have shown that incorporating curvature and tissue geometry dramatically changes the resulting patterns, the resulting Turing pattern. It actually better explains either branching patterns. In the case of lung epithelium, there's work from Dagmar Reiber that shows that incorporating curvature better explains the branching pattern in the lung, and the same for crypt organization too. And thinking about curvature in these are models on the right, but this has been experimentally tested too, that the ligands that are trapped in these crypts or buds do do experience the effect of geometry in addition to just this reaction diffusion interaction. So geometric feedback is important. The third kind of feedback, which is slowly emerging in the field, it's one of those things that is so obvious, but it needed a framework. That's how I see it. Is just changes in topology that are happening throughout development. So I gave the example of formation of a tube, either through folding and fusion, or through just cavitation. In this transition, um, it is a topological transition, and this surface is fundamentally different from this enclosed surface that can now have either a lumen that is pressurized. It can have fluid flow that can cause shear forces. So just having an open versus closed configuration, or a change in topology can change what kind of forces or signals the cells that are surrounding are going to experience. In another example, I'm going to go in a little bit more detail about this, if I have time. Is this organ that we study in the lab, is the formation of semicircular canal. Um, it starts off, actually, the first topological transition I haven't shown here is just cavitation of a group of cells that make this ellipsoidal structure. We're only looking at half of that ellipsoid. And then this tissue undergoes massive remodeling at 6 locations. I'm only showing 2 of those locations where it transformed these cells into buds that extend and fuse. So they are undergoing a topological transition to demarcate a donut hole or a torus around them. And this we've shown. This is unpublished work, and if I have time, I'll show you later. We can show that this contact formation allows for the activation of certain pathways, transcription factors. Because this contact happened, these cells were not seeing each other earlier, but because they came into contact, they are now able to activate a receptor that is required for the fusion of these two buds. So just summarizing that this is sort of the view that that we've all came to accept as a community in developmental biology, and of course, amazing work by folks that are there in CMSA and all over the world. We now know that this is reciprocal flow of information, where the remodeling of tissue or deformation of cells is feeding back to the inputs that are driving those dynamics. Another principle that I want to highlight that is, again, it's connected to the feedback, but I want to distinguish between the two is that morphogenesis is unfolding within constraints, and these constraints can be physical, geometric, or topological. So we can think of feedback as essentially how a system is evolving. If there are deviations, there can be feedback to correct things in place, and we can talk about that robustness too. But in contrast to feedback, we can think of constraints as what forms are even possible, what shapes are even possible or accessible during development. And here again, I'm speaking to a choir. We know that there are physical constraints in the system. So the way a tissue would buckle depends on its material properties. It depends on its stiffness. It depends on its bending rigidity. The way a tissue elongates depends on its viscoelasticity. So on short time scales, these most epithelial tissues behave as elastic materials, and on longer time scales, they behave as viscous materials. So whether a tissue stretches or recoils depends on the time scales on this force application relative to its relaxation time. And finally, this is something I didn't, a process that I didn't introduce in the first part on the many different kinds of morphogenesis you can have, is sorting of cells. But this is, again, it's this feature of cell sorting where the governing constraint is interfacial tension, and force balance is used a lot during morphogenesis. So cells would rearrange, for example, if you have a group of cells with very mixed group of cells with very different adhesive properties, they can rearrange or sort until the tensions at their interfaces balance and until the entire interfacial energy is minimized. So one can think that in these developmental processes, these physical constraints are really important to define which shapes are even possible. Now, the other constraint that I think is very relevant, again, it is very related and ultimately going to impact physical forces, but we can still distinguish it is geometric constraint. And this is coming from the shape, the curvature, the size of the tissue. And there aren't that many clear examples to think about this as many as we could for physical constraints. But one example that it's a recent paper, I think it's from last year. It's this beautiful work done in Drosophila hindgut primordium. So using both wild type and here there are some examples of mutants. The authors show that this hindgut here, it sort of grows anteriorly during gastrulation. So this and it has this what they call a keyhole shape. This keyhole shape essentially can be defined with this minimal model in which shape changes emerge from forces exerted by these actively remodeling tissues. But a critical component is this ellipsoidal geometry and the curvature at the posterior end. So just with these two features, this component was extremely important. They could explain the different shapes of the hindgut, and one can be observed in other species depending on the curvature here. Okay. The last constraint that I want to highlight, again, I think there isn't enough about this, so I've just abstracted the problem. So is topological constraint. So it's just a restriction that I'm imagining that's coming from connectivity of the tissue, not from its material properties, not from mechanical forces, but or the magnitude of forces, but just how the tissue is connected. So if you have, for example, a spherical shell, it has it let's say it has uniform material properties. If it has fluid within, the pressure is balanced symmetrically across the surface, and now it undergoes a topological transition into a torus. So if nothing else changed during this process, then you can still imagine that the pressure within this is going to change because it's in a torus. There will be an inner curvature and outer curvature. And the way the stresses are distributed are also going on the surface, on the cells, is also going to change. And of course, this constraint coming from this change in topology is going to directly feed back onto the mechanics of this tissue if it's undergoing morphogenesis. So, and I would love to have discussion about more such examples where topology constrain the kinds of shapes you can get. Um, how much time
Akankshi,
do I have?
can you hear us?
Yes, I can.
Yeah. So I mean, another I think beautiful example of where topology constrains morphogenesis is in hydra formation, where the you have topological constraint of putting a pneumatic order of the act. This is from Kinneret Karen's work, right?
Yeah.
The labs. Putting pneumatic order on a spherical topology, then you have to have these net disclinations in the alignment, and this gives rise to sort of anterior posterior axis patterning, and other work on the tentacle formation followed from that as well. Maybe it's a bit different than what you're pointing out because there's no topological change in the genus of the surface, which is clearly you're imagining your inner ear topological changes here. But I think they're both in Kinneret's work and also in some sort of backup slides that I showed on Monday, that we have these sort of topological constraints in a system with a fixed topology that play into the geometric dynamics or that constrain the geometric dynamics that are full.
Yeah, when I was searching about this, I saw all the work in Hydra, but I was just limited by my expertise in understanding. So I would love to just have someone talk about that, hopefully in one of the days, or we could have a one-on-one conversation about this.
Sure, yeah.
Vanessa, did you want to?
Yeah,
Yeah.
I was thinking, I also find the topology question quite interesting, and we've done some work with cellular topology, right? That is connected to the
Yeah.
kind of. So are you also thinking of constraints in that sense that if you have a tube, also your cells can be arranged or rearranged in a certain way that it's not necessarily going to happen if you have a flat sheet, right? Or a sphere.
Yeah, absolutely. And that's something sort of like, I mean, in my mind, it's hard to even disconnect topological feedback versus constraint. And
Hmm.
that's sort of what I was trying to allude to in the topological feedback part, that by the virtue of making and breaking contacts, you can allow for certain processes to happen or not happen. You're not going to have notch delta signaling if the cells are not in contact. In our case, we see a GPCR that's not going to get activated unless it sees a partner cell that is not in contact earlier. So yes, we think about it in that in at the level of cell connectivity as well. But here I was trying to imagine more at the surface topology scale
Hmm.
on how stresses would redistribute once you change the topology.
Maybe
Yeah.
another just example to throw in here is that Konstantin Dubrovsky has some great work on how the spherical topology of the gastrulating fly causes very different global constraints on the dynamics than you would have if you were unwrapped into a disk.
Hmm.
so that because
Mhm.
elasticity is long range and because you have a periodic boundary condition, then your
Yeah,
tissue
I
reduces
forgot to
itself.
mention thanks for pointing that. I forgot to mention both in the geometric constraint and topological strain, the boundary condition. And this is something like you introduced really well in your talk. So yeah, absolutely. Um, I could entirely skip the next, I mean, or I don't know how much time I have. It's close to 12, so I can maybe take 5 more minutes. Yeah, I feel like yeah, I maybe I talked really fast, but yeah, thanks for interrupting me because it's just maybe too much talking. Okay. So I just want to transition um to this idea that I'm not sure if I can call this as a principle, but this is something that I think we as a community ask a lot on what makes development robust and reproducible. We know that development is robust to both intrinsic noise and extrinsic um, insults, environmental perturbations, variations, and so on. And I've tried to sort of abstract it as this idea that development is robust because we have constraints that are canalizing the possible shapes that tissues can acquire. And this is something that Noah and others have also alluded to in their talks. And development is robust because you have feedbacks in place that are correcting errors that are happening during development or that are buffering variations. So if you have positive feedbacks, you can amplify small signals, buffer variations in initial signals. If you have negative feedback, uh, you can prevent overgrowth and so on. So because you have these feedbacks in place, you can make more development robust. And we think about these questions using this organ that I already introduced, the semicircular canals. I didn't quite tell what it does. These are three tubes in jawed vertebrates, and there could be one or two in jawless vertebrates that are within our vestibular system in the inner ear. They help us sense balance. So when we move, there is fluid moving in these tubes that is being transduced by hair cells that sit at the base of the tubes. And I'll introduce this in more detail next week. But what I want to highlight, and as I said, we study them, we study their formation using zebrafish. And this is something I already mentioned too, that the beginning of this structure is happening from this formation of vesicle. In zebrafish, this happens with cavitation. In chicken and mouse, it's happening through this folding to make the otic cyst. But essentially, you're making a hollow vesicle. It's not really hollow. It's filled with very salty fluid that drives its expansion. So it's a single layer of cells around. It's a tight epithelium, and they're not dividing very much. They are, but not enough to explain the expansion. It's being driven by hydrostatic pressure. And once this tissue reaches this certain size, it remodels dramatically to give rise to the semicircular canals. And we'll observe this remodeling together. So here is a 3D rendered view of that tissue. It has this invisible lumen. I've just marked here for reference. You can just morphologically distinguish it with this very ellipsoidal shape. And its morphogenesis begins when six regions in this epithelium on the top half of the these buds then fuse to one another. So this is similar to how the neural tube was closing in. Um, and then this fusion, I'll just make this faster to save time. This three fusion events, so they're happening in the anterior region, posterior region, and later on in this orthogonal plane in the ventral region. this fusion results in the formation of three pillars. And the tube that they are demarcating around correspond to the semicircular canals. So here you've looked at this illustration before, and it hopefully like you can connect the confocal images or the time lapse with this illustration. So here I've shown two buds. They extend, fuse to form pillars, and they demarcate this tube around. And this is what an attempt a postdoc in the lab made to show what the tubes would look like once those pillar form. So these are the three tubes. They're roughly orthogonal to each other. And one thing we observed by collecting a lot of time lapses of this process is it's highly variable. So here you can see that this is a classic mode. The buds approach each other and they fuse. They scan a little bit of space around, but they eventually find each other, go through this fusion process, and ultimately form pillars. In another example, you can see this auric vesicle has expanded more than average. And if we quantify how much buds need to extend to find each other, they would need to extend a little bit more to counteract this expansion of the auric vesicle. But they still reliably meet and fuse to one another. In the third example, I don't know why it's not zooming out. It's supposed to zoom out, but in the third example, we can puncture this tissue, causing it to collapse. So it loses some of its pressure. Now this bud is going to meet at a very obscure angle, a very acute angle with its partner bud at a very acute angle, but still be able to form a pillar reliably. I'm not showing the endpoints here, but the endpoints are remarkably robust. So you can start off with very, very. Is there a question?
Akankshi?
Yeah.
Yeah, just quick question. What's mediating the adhesion of the columns to each other as opposed to sort of sticking non-specifically to other structures?
Yeah, really good question, and I'll keep it short for today. But short answer, we don't know. Long answer, I mentioned this specific GPCR. So there is a mutant in the field of this GPCR where the buds grow past each other. They don't stick to each other once they find each other. So we think it's something specialized about this GPCR. We don't know what its ligand is, if it's mechanics that's making the cells sticky to be able to result in a successful fusion. And to your point of like, what's preventing them to stick to random surfaces, we think that's physically, like speaking about physical constraints, we think that space is that probability is very, very low. That given the initial position from where the buds are growing to them finding each other is constrained by the size of the auric vesicle, its aspect ratio, the sort of roughly longitudinal extension of the buds orthogonal to the plane where they're starting from. So given these constraints, the buds are likely to meet each other, but they meet each other at many different places. So this is one of the things that I wanted to highlight, is that we can categorize variability in many different ways. And I'm going to talk about bud extension in my research seminar next week, but I can highlight the site of fusion, which is connected to your question, Alain, where they can meet at many different places within the buds. And it doesn't matter where they meet. As long as they meet, they express this transcription factor at the site of fusion that is required for successful pillar formation. So I don't know if this addresses somewhat your question. But we think that because the expression of this transcription factor is dependent on the contact of these cells, this topological transition or this formation of new cell-cell connection is what's canalizing the variability on the many possibilities where the buds can find each other. Instead of predetermining a small subset of population that can fuse to one another. So in another example, I'll go over this very quickly because it's like an entire body of work that it'll be really hard to summarize, but I still wanted to show because it's really beautiful on how the variability is getting reduced. So I mentioned that the aortic vesicle is expanding, and the rate of which at which it expands, we haven't quantified it yet properly, but if we just observe qualitatively, we know it's different between different vesicles. And I told you that the buds can meet each other at many different locations, which results in many different shapes of the pillars we observe. So the buds can approach each other at many different angles, and they still fuse, and they can form pillars that look all kinds of shapes. Like they're not straight, they're bent, in many different ways. And over time, what we observe is the pillars straighten out. And this is in conjunction with the overall expansion of the aortic vesicle. So now we're doing experiments, and we have very promising data that supports that this straightening of the pillar is happening from this mechanical expansion of the aortic vesicle. So you have these bent configurations initially, but over time they're getting straightened out because you're expanding the entire epithelium. So it's all I mean, these pillars are in continuum with the rest of the epithelium. So this is, I just wanted to I mean, this is the last slide I have. So I just wanted to bring these as discussion points on thinking about variable trajectories of development that get canalized by these constraints or feedback from different sources. Yeah, thank you all. And if there's